Hello, hello, and welcome everyone to Soil as a Living System. Lessons from Dr. Elaine Ingham in a decade of practice. Today, we're going to hear from Nick Tomasini, and we are on this webinar series, Soil Food Web, A Living Legacy. This series is happening since October the 3rd, and we will end with a celebration of Soil Day on December 5th, where we're going to have a special edition. We're going to see in a couple minutes what that does means. Do not forget that those webinars are happening 9 a.m. Pacific time, so please adjust accordingly to your time zone. And as always, if you need help, please do not hesitate to contact our amazing team for any support. I am Dr. Carlo Portugal, your host for today, and I will be guiding you through today's discussion with our amazing panel, which I have the honor to be joined by Nick Tomasini and Evan Buckman. Nick, would you please introduce yourself for our group here today?
Sure. It's a pleasure to be here. My name is Nick Tomasini. I own and operate Humankind Oregon out of Bend, Oregon, where we, of course, focus on soil food web consulting, agronomic recommendations and planning, and some nutrition management, soil biology assessment, and compost production with my partner, Robert Fair, over in the Willamette Valley, just outside of Eugene. Again, it's a pleasure to be here, and I look forward to getting into more about my journey here in a little bit.
Yeah, I have the pleasure to be the host on a webinar with my neighbor. I'm also in Oregon, Corvallis. I just know that Nick is a fellow beaver, so go Beavs. And I have the honor to be the science lead and also instructor here at the Soil Food Web School, and also with my consultancy company, Soil Domains, which we work in Oregon and Brazil. And I'm delighted to be a co-host with Evan Buckman. Evan, would you please introduce yourself for us today?
Hey everyone, my name is Evan Buckman. I'm calling in from Austin, Texas, and I'm the new executive director of the Soil Food Web Foundation. Really excited for today's conversation with Nick. Nick was actually one of the first Soil Food Web practitioners that I met early on in my journey of getting to know the Soil Food Web as a movement and a body of work. So really excited for what we have to share today. And I see a bunch of comments already in the chat. If you're here with us live, please let us know where you're calling in from. Always love to see folks connecting from different parts around the world.
Yeah, they got me, Evan. I was just like trying to invert the order, but I guess everyone already knows the drill, which I love. Thank you so much, everyone. We are still waiting for people to join us from Antarctica, so one day they will show up. So, oops, sorry. Okay, so let's just recap what we had so far on this series that we are celebrating a living legacy in a five-part webinar series. So the first webinar was Lessons Hidden in the Ecological Monograph, where we had Daniel and Adam talking about their experience and how they came along with the soil food web approach, targeting especially Dr. Elaine's paper from 1985, when she really dive in, in the soil food web knowledge as we are continue to learn today. So they went through the 5 key insights in how soil microbes drive the plant growth and ecosystem resilience. It was incredible. Webinar 2, we had a conversation about generations of soil food web practice across landscape, where Todd Harrington, Vivian, and Joe Tobias just walked us through of decades of experience in soil food web practice and application. They also expand a little bit on the new frontiers of soil regeneration. Another webinar that so many layers, so many information that I'm still trying to digest everything we heard from them. The last webinar, the previous webinar, we heard from Celine how soil as a national defense and how we can rethink soil as a strategic resource. Celine did a great job examining the soil diversity and how this is interconnected with agricultural productivity, food security, territorial resilience, and how this can impact global challenges. It's a very, very deep food for thought on her webinar. And as always, you guys can reach our YouTube channel to rewatch any of those webinars. And you can rewatch as many times as you want. I'm one of those that go back there time to time to not just get inspired, but also to learn from them everything that we can. And today we have the pleasure to hear from Nick Tomasini, how soil as a live system and the lessons learned from Dr. Elaine in a decade of practice. Nick is the founder of Humankind Oregon, a certified soil food web consultant, independent organic farm inspector, and he will go through his resume for us. But above all, he will explore how the soil food web provides this powerful edge for farmers and practitioners managing high valued crops. We're going to hear much more about how to sustain an ecosystem service and adapt capacity. And we cannot forget to celebrate what we do here the most, soil health. That's one of the reasons why we are here together. And this year, to celebrate World Soil Day, we're going to have a Soil Food Web Community House Party. If you join us in the previous sessions, you know how fun this is. We're going to have about 15 themed breakout rooms where you can jump in from one to another as you see fit, and share everything with our community. This day is about you, is about how you are helping to bring up life back to soil. We wait for you guys there. Do not forget that's 9 a.m. Pacific time, 5 p.m. London time. Adjust accordingly for your time zone. And we went through the introductions, and we're going to have about 45 minutes on the presentation by Nick. We're going to hear a couple minutes from Evan. What is the news in the offer we have now? And we're going to jump for the Q&A. Do not forget to put your questions in the Q&A chat on Zoom. And without further ado, Nick, take the floor. You're muted, Nick.
Sorry. Again, it's an honor to be here. And I would just wanted to start out by saying a few words about our friend and our teacher and mentor, Dr. Elaine Ingham. We're here to celebrate her 4 decades-long career that began with 2 decades in academia. Her greatest contributions, though, came from stepping beyond it, building a framework that unified soil biology, ecology, and practical agriculture for all. Her legacy is carried by the thousands of farms and projects and gardeners and practitioners that she has inspired to now place biology at the center of their decisions. She authored and co-authored key works in our understanding of microbial interactions that led to a basis for grouping microbial grazers into functional groups, microflora into broad guilds, and the trophic interplay that connects them, all of which she taught in a way that anyone could understand. She's always been uncompromising and unapologetic in her deep convictions to understand and support soil life, but behind that resolve was a genuine desire to help people see the living world beneath their feet and use that knowledge to heal their land. So thank you, Elaine. I first became aware of Elaine as an undergrad at Oregon State. She did most of the research at the end of her career in academia there. And I think she was there until about 2001, and I started a little after that. And then it was another year before I switched over to horticulture and agriculture as my area of study. Um, and so, the curriculum that I learned there was important, but it was very conventionally based. Um, and there was certainly something missing there for me. I was interested in learning more about sustainability, which was a big term and topic at this time, and organic farming, but I wasn't getting a lot of this curriculum, at least from lectures and professors. So I kind of had to seek it out on my own through classmates, the organic growers, through the Organic Growers Club there at Oregon State, which I think is still an establishment there and a great spot. Um, and I have to tip my cap to my friend Ian Hunter, who was the first person to actually point out Elaine, bring Elaine to my attention. So he and I would have, yeah, we would have long discussions about soil biology and biochemistry and biodynamics, which he was really, really big into and still is. And yeah, those were very important discussions. I met more and more people who were also interested in these things. And I think there was a general buzz around Elaine and her work um, through from a lot of those students, regardless of their background, if they came from multi-generational farms. There was a buzz around it for sure. And luckily for us, Elaine had Soil Food Web incorporated in Corvallis at this time, which is where OSU is. And so, we were able to go there and learn about soil biology, buy compost. She was selling Alaskan glacial compost and fulvic acids and manuals and information. So that was just a major resource. And so we were using those tools. We're learning about those tools, buying that compost, putting together these little DIY brewers and starting to learn about this stuff. So these are actually from my library. Some of these books, especially the tea manual there, I've had for about 20 years. And I spent a lot of time in that book early on and sort of going back and forth. I didn't have FaceTime with Elaine at Soil Food Web Inc., but we would go in there and whoever was working there, they would sort of give us the lowdown on how to make teas, on what was going on with this compost. He would sell us like small cardboard boxes full of compost that we could use, these special fulvic acids and kelps and stuff. And so we were getting pointers there for sure. But the soil biology primer in the middle, actually, if you go to the USDA NRCS website and you go into their education resources, it remains the primary Soil Food Web education resource for the Natural Resources Conservation Service, and USDA as well, links there. So that was a big publication, and it really helped to put things in perspective for me. It's very approachable. It's not a very long manual. There's great pictures in there. There's absolutely great content. It's where I started learning about ecosystem and microbial succession and how those are interlinked, seasonal fluctuations in microbial activity, what types of microbes exist in different types of ecosystems, these microbial guilds, and functions, and also photos of these microorganisms. So that today, I think, remains just a great, primer as it's titled. And yeah, you could still buy that off the NRCS website, as a matter of fact. I think that was actually published by the Soil and Water Conservation Service in 2000. I think around 2000 is when that came out. Anyway, then of course the tea manual, which I talked a little bit about, and learning about the fact that compost quality is absolutely critical, factors affecting compost tea quality, recipes, what some of these desired minimum microbial ranges were that she was working out at this time. And yeah, it just was very eye-opening for me, and certainly as I went through my undergrad, changed the way that I thought about soil and ecosystems and farming, which I was very interested in. And then, of course, on to the qualitative assessment of microorganisms manual there, which came out. I think that was like 2005 or so, and I got it right around that time, maybe a little after in 2006. But bought a dinky $50 microscope, started sort of looking at these teas and trying to identify organisms. The microscope I had was very low quality, very blurry. But we were trying, like how good were these teas? We were seeing these great results in our gardens, but we wanted to go a little bit further. So I actually I should have waited to go to this slide, but after my undergrad, I went to work for a very big farming operation there in Corvallis as an integrated pest management field scout. And while I was there, the head scout for this farm, I mean, this was a massive farm. We're talking 150, $160 million a year gross processing facilities that ran 24/7. Some organic ground. They were Food Alliance certified, which was sort of the sustainable certification where there could be no residual pesticides in any of their produce. So I became the field scout for this, and then the head scout left. So I became the interim head field scout for this giant operation right out of college, where I was scouting 25 to 50,000 acres of crop per month. And it was it was a massive undertaking. It was very eye-opening. I learned a ton. It was a good experience for me. Um, I basically would go out and do insect counts and look for disease pressure and weed pressure, and I would submit a daily scouting report. And anytime an economic threshold for pests or disease was breached, um, they would intervene chemically. So they had some organic ground, as I said, they were Food Alliance certified. That did not mean they didn't spray. And so I found myself on one or two occasions entering a field that had been sprayed with malathion that morning, and I didn't get the email. I entered the field from a different side, so I didn't see the sign at the edge of the field. So I found myself in the middle of a recently sprayed field, and like I said, this happened a couple of different times, and I started to become dismayed by being in these fields. And for the most part, these fields were not being sprayed. But again, when these thresholds were breached, they would be sprayed, and there was generally good communication, but there was still exposure there. And so after they hired a new field scout, I worked in their microbiology quality assurance department for a little bit, but soon after, I was looking for something new. And so I basically packed up my skis and my mountain bike, and I moved out to Hood River, and I went to work for the Oregon State University Mid-Columbia Research Agricultural Research and Extension Center. Yeah, that's that acronym. As a research assistant for a soil scientist, Dr. Shinhua Yin. And yeah, that was a great experience too. We were working on tree fruit, we were working on ground management and different pruning techniques and fertility and all these kinds of things. And yes, there were still chemicals being applied, but yeah, I was learning because the scientist I was working with, English was his second language. I was doing a lot of writing as well. So putting together tables, doing some of the writing, managing some of these trials. And there was actually soil biology microscopy data included in some of these trials. So it was kind of pulling me back into that way of thinking about things, and just seeing how some of these different treatments we were using were affecting soil biology in whatever way. But so that was a good experience for me. And so at that same time, I had the opportunity with a friend to lease some land in the Upper Hood River Valley in Parkdale. So we spent my wife, I wasn't even going to include these pictures actually. My wife and I were going through some old photos and we found all of these, and it was just, I hadn't looked at them in 10 years or something. So thought it would be cool to put these up and show people kind of what I was doing back then. So, we got this farm going, and I was making trips back over to Corvallis to buy compost from Elaine so that we could apply compost tea in our greenhouses to our fields. We were practicing all of these things we had sort of aspired to and been learning about, cover cropping, crop rotation, using sort of like living mulch in our alleyways. And yeah, so this is back 2007 to 2010. I was doing this in Parkdale. And yeah, I was sort of cutting my teeth on the farming side of things and using more of this soil food web knowledge from Elaine. And, it just pulled me more into the farming side of things, more interested in soil. And so at this time, we ended up losing the lease on this. There were apples here too, and those were being transitioned to organic. So we were helping out with that and doing our CSA, farmers markets, restaurants, anywhere we could sell produce, and we did pretty well. But they ended up retaining control of this section of their farm that we were farming on and putting cider varieties in there, cider apple varieties in there. And I was looking for the next thing. So I applied. There was finally an ecological agriculture program at Oregon State. And so I looked at that very closely. But again, I remembered my undergrad experiences, and I thought about, what I could actually get into as far as research, and if I could actually continue to do the things that I wanted to do, that I wanted to learn about, that I would want to research. And I had my doubts. I also wanted to be a farmer, and I was just a little concerned about taking on more student loan debt to, become a farmer and be unsure about how much money I would actually make, how difficult it might be to pay back all the loans that I already had and taking on more. So I learned a lot about, I knew about Oregon Till Certified Organic because it's also headquartered there in Corvallis, Oregon. And I did try to go to work for them right after college, which didn't work out at the time. But I thought to myself, how do I get on as many organic farms as possible to see what's going on? So I did my training with the Independent Organic Inspectors Association in 2011 and got my certification to be an independent organic inspector. And I went to work for Oregon Till Certified Organic as a contractor. And I was there from 2012 to 2020, and I did over 550 farm inspections for Oregon Tilth during that time. And I saw a lot. I saw many organic soils that were not in good shape. Sort of to my surprise, I was still learning about what was actually going on in agricultural soils. I wasn't super proficient with the microscope, so I was still making a lot of assumptions, like, oh, a lot of the compost is good. It's not, just this compost I can get from Elaine. Oh, people are using compost. They're spraying, applying teas and things like that. So these soils must be alive and very productive. But that wasn't actually my experience on a lot of farms. So yeah, it was more of, there was a lot of heavy disturbance. There was on a lot of farms, a lot of tillage. Compost seemed to be almost falling out of fashion during this time. So I was seeing less and less of that. I was seeing cover cropping not really working out for farms. Maybe it would be sown, but that didn't make a stand, and then people would eventually kind of move away from that. So I thought about it as sort of this conventional organic approach as I experienced more and more of these farms, visited more and more of these farms and learned about them. And when you're performing a full audit of crop and financials on these farms and going through all of their practices, you really get an idea of what's going on out there. and why the farms that are making it work are able to make it work, and why many of them that had low gross sales were sort of struggling, were not necessarily making it work. And so it was just, I don't know if I could have got that in grad school, frankly. All of that, all those discussions, seeing all those farms and getting into all the details of what they were doing. And what I saw from the most productive farms was that they were, they were very proficient with managing their fertility and their rotations. They were using precision agriculture with regard to nutrition and mineral balancing. Um, they were applying compost, and they just, even though there was still heavy disturbance and a lot of successions, they were making it work. And so I paid very close attention to those farms, and that sort of brought me back into sort of the Acres USA world, which I had studied a lot as well as I was learning more and more about soil. And the Albrecht papers, for those of you that are familiar with Dr. William Albrecht and his base cation saturation ratio approach, or mineral balancing approach to agronomy. So I was getting more and more into this and learning how to integrate this with the biological principles that practices that I thought I was proficient with. So I was doing a little bit of advising and consulting with a couple of operations on this front early on, and I yeah, I was still learning, how to really make soil work, what it was all about. And I think there were a lot of missteps during that time as I was trying to figure it out. And this was sort of perplexing for me. I think a big part of what was going on was I was making assumptions about compost and these sort of soil food web practices that I was trying to implement from the early days that I'd been practicing myself, but probably not using great compost, I think is what it really boiled down to. So I started Humankind Oregon back in 2015, where we were doing some of this consulting, and I was just I was still wearing my inspector cap, and I was doing a little bit of this as well. But then I I did my soil food web. Well, I should tip my cap to Scott and Sarah of Crescent Soil, because it was through social media that I met them, and that's when I realized they let me know that they were training with Elaine, learning how to use the microscope, and it was clear to me they were more proficient than I was on this soil biology side of things. So it was really them that prompted me to. It basically made me aware that I could train with Elaine, and so I started doing my education with Soil Food Web through the Environment Celebration. And I became a consultant in 2016 through that program. And at that time, Elaine and Dr. Caroline Rollins were the only mentors, I believe, there. So Elaine was actually my mentor. And so it was great to finally be Elaine and Caroline. It was great to finally have FaceTime with Elaine and be able to pick her brain and to learn from her directly, and to go through all of the training, making the compost, making the teas, how to apply stuff, just to be able to ask questions and get really good feedback from her at this point in time. So that was all very, very pivotal for me, and really changed the course of my career and my ability to be effective as a consultant and somebody who understood soil much better, and could make it work for the farms that I was working with. There we go. So I'll just share a couple of large scale projects that I've worked on in the last couple of years. Um, there have been a lot of operations, a lot of crops in Oregon, the Northwest, and beyond that I've worked with. And projects have gotten bigger and bigger over time. And, some of that mentoring from Elaine was in a professional capacity as well. What does it look like to work with these large operations? Um, how to, protect them and their data. And yeah, sometimes, I'm under NDAs for some of the projects that I work on now, so I can't share everything that I currently work on or have done. And even some of these, I don't have full permission to share everything. And Elaine's advice was always, get the data, get the data. And now I understand more than ever why she was saying that, because it's very difficult. And I understand because they have these larger operations, they have their own internal IP that you have access to, and you're doing a lot of work for them and adding to this IP, and they don't want you to just go to their competitor and basically divulge all this information. So I completely understand why this is the case, but it certainly makes it difficult to share your work and to promote yourself with, let's say, white papers, that kind of thing from the work you've done. That said, the networking aspect has still been great. So even though I don't share a lot publicly about my work, I've been able to network through my clients and get new projects and um, projects through Elaine over the years. I've worked on projects with Elaine. She's given me speaking opportunities. Yeah, she's given me a lot of at bats for sure. So I'm very thankful for meeting Elaine. Definitely my most important professional mentor. Anyway, so Great Basin forage and silage. This is a very, very large operation. They probably have about 10,000 acres of irrigated land. We worked on about 1,200 acres of those pivot irrigated fields. But beyond that, they are hundreds of thousands of acres of flood irrigated meadow to long-term leases through the BLM and National Forest. They are just a massive cattle operation, and so it was great to work with them, and definitely eye-opening for sure to get into learning about some of their barriers and some of those issues. So yeah, I mentioned about 1,200 acres is what we worked on, and those soils were sticky clay to sandy loam, about 4 to 5% organic matter on those fields. So quite a variability there. They were running into productivity issues where they couldn't even grow staple crops for them, like alfalfa, that they were cutting hay for and feeding to their cattle. They had compaction issues, heavy compaction in some areas. They had some maloidogine or root knot nematode issues in some of those fields where they could not produce alfalfa at that time. And so, they were looking for another way to do things, a more regenerative approach. And yeah, just a little bit more about this area. This is in the Great Basin. It's high desert. So about 9 to 12 inches of rainfall here annually, and a lot of that is snow, as a matter of fact. And then only about 90 to 120 frost-free days in this area. So started talking with them in 2021 and sort of making a plan. And what that kind of looked like in a nutshell was some prescribed cultivation at the outset of this work to address compaction issues in some of these fields. Some of that compaction was so bad we were unsuccessful in even like being able to get a ripper through there. Penetrometer readings were just off the charts. I don't even know what they were. They were off the meter for the penetrometer. So but that's where we started with a lot of these fields to kind of address that. And then we immediately came back with these very diverse forage mixes that we kind of termed regenerative forage mixes. So these were the backbone of basically working on getting diversity out there, working on soil health and trying to enhance their productivity or bring it back to where it once was. So we started sowing these with a no-till drill, and that was sort of another way for them to get away from using herbicides to spray off these fields. We were applying compost extract at about 20 gallons an acre for about twice a year through the pivots. So we were injecting extract right into these pivots, and then they were grazing down a lot of these regenerative mixes to use cattle to help regenerate, the soil health here. And then, yeah, I mentioned the no-till seed drill already, but it was, these ranchers are super innovative. They didn't have time to make compost extract by hand with individual bags. So, we sat down one evening after the first day out there, and we basically devised this plan to get this giant cattle trough on the back of a flatbed truck, about 1,200 gallons. And we would load it up with compost, and we would literally take shovels, and that's how we would do our extraction. So we would get a big vortex going in here. I'd look at it just to see, sort of what the biomass and population levels were. Were we making a biocomplete extract? And then we would filter out of this into our tanks that we injected from. So it was funny. We're like running around out there with these totes of compost, this giant cattle trough strapped to this flatbed truck, and we would, we would basically load these things up, make extract, set that pivot to run and inject the extract, and then we would drive on to the next one. And so we could set up 3 or 4 pivots in a day to just be spraying out extract, and then we would do the same thing the next day. So anyway, objectives here, they wanted to regenerate their soil health. They needed to improve their yields. Yields had slipped. They couldn't even grow a lot of these crops anymore. And their feed quality was slipping. Winter carrying capacity was going down. There wasn't a lot for these cattle to eat out on the meadows during the winter months, so they did rely on feed for that. They also wanted to add some new crops into their rotation and add just some more diversity. And then ultimately to reestablish productive perennials, mixed perennials they could be grazed, they could cut hay from it, but they would basically sustain soil health in the long run. So here are a few photos of these are actually, this is a process that started in 21. These region mixes that I mentioned were grown. Some of these fields did not bounce back as fast, the ones with the heavy compaction. So there were actually 2 years of that sort of region approach, which was basically these 10-way mixes, compost extracts, some biostimulants in there as well, and then grazing was sort of the approach to regenerate those soils. And so what we saw, well, I should talk. So we did implement a new crop, which ended up being non-GMO silage corn. And I have to say, I did not see the silage corn coming. There was some popcorn that we used in one of our regen mixes that had done really well. And this operation previously did not consider corn as a viable crop for them. They didn't think they could grow it in this region. Where they wanted to plant it was actually in one of the fields that had these maloidogine nematodes that I had found, and so I was warning against that. They did just fine. County average is between 15 and 20 tons per acre on silage corn in some of the lower areas where they do grow corn in this county. And so they were able to achieve 15 to 18 tons per acre on their silage corn, which they were ecstatic about. And I was happy they were able to meet their goals and add this crop. And, from their point of view, this was going to help them, their winter carrying capacity, finishing cattle a little sooner, putting more weight on them, that kind of thing. So that was a goal of theirs, and they were very pleased with that. The mixed perennial fields, alfalfa, timothy, we were able to add some other forbs in there too, some plantain, some clover, some burnet, just to get a little bit more of a taproot in there. Some of those are also very good for cattle health. So they were seeing really good yields after regenerating these soils of 3 to 4 tons per acre. And so basically, it became a no-till rotation where they would do their regen forage mix, the extracts, the grazing. They would go into silage corn, perhaps multiple years of silage corn on the same field before going into perennials. So again, I didn't really see that coming. They were very happy about that, and so I helped them reach their goals. But maybe I had sort of lofty visions of grass-fed and always grazed and never needing to cut. But they carry a lot of head of cattle here, so some of that was a little bit outside of the context that I could really understand. So I would say, wins here, going no-till, improving diversity and getting those forbs and perennials back into a productive state, eliminating herbicides, so not spraying off fields before planting a next crop. Um, the application of biocomplete compost extract, and they were just really happy about having that solid regeneration protocol if they ever slipped again. So for context, I think continuous corn is very tempting for an operation like this, at least on some of their fields. And once they realized they had these regen protocols, provided them with a tool to sort of course correct, perhaps in a cyclical pattern, meaning, if and when soil health slipped again from something like continuous corn, they knew how to navigate that. So, um, ideal next steps, I think, for this operation. And I'm sort of in their ear about this all the time, but I'd like to see an injection system on that no-till drill so that we could be putting compost extract in furrow as they're seeding. Um, yeah, a more precise nutrition program beyond just NPK. For their silage corn, they do use 80 units of urea to make that yield. So more traditional or more conventional or standard for that would be probably 150 to 200 units of nitrogen, pounds per acre of nitrogen. So that was another compromise in this project, but as far as other agronomists in this area were concerned, that was of importance if they wanted to make yield on corn. So a little bit of a compromise there from my more idealistic point of view, but in the real world, that's what these farmers felt like they needed to do, and so far they've had good success with that. And then I would love to see composting equipment on this operation. There is a lot of manure. Most of it is out on range, out on pasture, out in the national forest, but they do collect a lot of manure in the wintertime. And so I would love to see a composting operation for them. And I've been pushing for that as well. But, I'll continue to do that. I'm just happy I was able to help them reach their goals. Good learning experience for me. All right, so I'm going to talk about this next project, which is a big tea estate in India in the Terai district. So this project was 100% remotely managed. I have not been to India. Um, we started with one tea estate that is 500 acres. It's been conventional for many generations. And yeah, there were a lot of concerns around making this jump to a regenerative biological approach. And so this is very much so a carefully managed transition. And yeah, so it's shade grown tea in the Terai district in India. Um, and and Elaine was involved at the outset of this project, so we did a little bit of, we had a lot of correspondence with the client in the beginning where we sort of talked about where they were coming from and where we wanted to go and how to get there. So there was a lot of skepticism around going cold turkey on certain things. Um, but we were able to stop across the entire 500 acres all herbicide, all conventional, all systemic fungicides and insecticides from day one, and that was in 2023, and that is still the case for this estate today. A little bit about the soils here. Most unique soil I've ever worked with. Silty loam soils, 1.5 to 2.5% organic matter here, and a pH of 4 to 5.5. And tea in these regions, they target a pH of 4.5 to 5. So that's kind of what I was working. They get 130 inches of rainfall here. So there's just a ton of hydrogen coming into the soil and acidifying this soil. So that it's already very acidic, but this is sort of where they wanted to manage things. And I've pushed on that a little bit as we've gone here, and I'll get into that a little bit more. So as I mentioned, yeah, we came to the consensus that we were going to immediately cut all synthetic fertilizer by 50% from day one. And basically there were only three in use. It was urea, single superphosphate, and then muriate of potash, which I had some concerns about continuing to use that at all. It's very caustic. It's detrimental to soil biology. I think it's one of the worst fertility inputs you can put out there that would have a direct impact on soil biology. So we've been slowly moving away from that and figuring out ways to put K out there through this transition process without using MOP, muriate of potash, excuse me. So again, as I said, 100% remotely guided. We've gone through 5 compost cycles, and we've met microbial targets on all of them. You can see in some of these photos that they were just meticulous with this compost management. I couldn't have imagined them following instructions or working any harder on this aspect of things. And it really shows in the quality of this compost. We've not only met minimum targets on every single one of these cycles of compost over the last couple of years, but they've been, 300 to 600 micrograms per gram of fungi in all their compost. For those of you that, are soil food web students or practitioners here on the call, you'll kind of understand what I mean by that. So excellent outcomes, really great micrograzer, nematode, protozoa outcomes in here as well. And so we immediately, we were concerned we didn't have enough from the get-go to do extract. Obviously, we didn't have enough to broadcast across 500 acres. So we started working very intensively with compost teas and a few of these blocks. And we've seen some good results there. We were able to, so we're working with teas at this point, making compost. We're shifting over to manure as fertility. They had existing vermicomposting, so we've been working on biological outcomes with their vermicompost as well. We've implemented fertigation and foliar so that we're not putting so much conventional fertilizer into the soil, especially in these more biologically intensively managed blocks. It's been great to work with different feedstocks, things like water hyacinth, which is invasive there, banana stems. We use tea prunings and chips from the tea estate, bamboo, cow dung. Those are basically the staples in the compost we're making here. So just to kind of recap from the slide here, we've gone through the composting, the tea, had to set them up with a microscope and start to train them on some organism ID so they could do some qualitative assessment. And just really astute, really organized, really became really proficient with that quickly. So we've gone from chemicals to IPM scouting as well. There are a lot of exotic pests and diseases that were also unfamiliar to me at the outset of this. So set them up with some scouting protocols so that we could kind of observe where hotspots were and also some biocontrols. So things like Metarhizium fungi. We cultured trichoderma from this tea estate, and we use that pretty broadly as a biocontrol. And we use some Bavaria bassiana and a few other microbial biocontrols, and we've had great success with that. Also, along the way, they started out by making, they were able to make like 90 tons of compost by hand on the first cycle there, which I thought was just wild. And now they have a turner, so composting is mechanized there, and they're able to make a lot more on each cycle. And we're still seeing just amazing results with that compost. So better than I could have expected. We've got some additional spray rigs out there for compost tea so that they can make monthly applications there. And as I mentioned before, we're fertigating. Oh, yeah. Well, we're also fertigating extract in a lot of blocks or sections on this farm, along with the manure and the new protocol of utilizing manure and minerals and a little bit of foliar nutrition as well. So this one, again, a very carefully managed transition. It's been a journey, but it's gone super well so far. Um, I think largely because of the incredible attention to detail. So let's just run through some of these results. And I pulled all of these directly from a client testimonial, which is on my website, because we're still, yeah, we're still, well, I'll get to some updates on this project in a second. But so now we have a successful composting program for this operation. So they are able to manufacture their own source of diverse microbiology for this regeneration process. We've established data-driven nutrition and biology programs. It was a challenge to find labs in India where we were getting good and consistent data. So we went through a couple of labs before we finally landed on one, and we're able to get really great soil and tissue data on this tea crop now, and that has been immensely helpful. So we are developing and modulating and calibrating that nutrition plan all the time, reducing inputs and really maintaining consistent yields all the while. And that this was just massive for them. Are we going to be taken out by a scourge of insects or disease as we pull away cold turkey from these biocides? And that has not been the case. We've managed to maintain consistent yields across this entire estate, not just the more intensively managed biological blocks that are receiving more compost tea. They've transitioned from chemicals to IPM scouting and biocontrols with great success. We've seen major reductions in pests, incidence, and pressure, even while neighboring tea estates were basically suffering crop failure from very, very high pressure from loopers, from tea mosquitoes, and some of the more common pests in this region. So that was very pivotal, I think, because that was a huge concern. Are we able to really drop all these chemicals and improve, basically maintain our yields and our quality and all that? So with um, with regard to quality, the way that tea quality is assessed and what actually sets the market price for tea in India is through through tea tasters. So they're professional tea tasters, and sorry, they're professional tea tasters, and when they did blinded side by side tastings of these teas, particularly our more biointensively managed blocks, they were hands down higher quality than the rest. So again, maintaining yields, managing pest pressure, seeing a resilience in crop they hadn't really seen before, and improving the quality over the course of these first couple years. And so I'm excited to report this project continues and is now expanding from 500 acres to an additional tea estate and 2,000 acres in total, where we're going to essentially we're going to follow the same roadmap that we did for this initial estate, and we're going to be able to do it more efficiently, faster, and on a timeline that we understand. So we are just starting they are starting their sixth round of mechanical composting right now. Our goal is to produce at least 200 tons in 2026. No, 300 tons of compost and 200 tons of vermicompost in 2026. So it'll all be produced in a central area. These tea estates are only about 15 minutes apart. And so, yeah, we're going to follow that same roadmap. And um, with the initial estate, we're going to continue to reduce our fertilizer input. We're going to continue to transition further into minerals, manure, organics, those kinds of things. And yeah, it's there was some skepticism for sure, and including from the tea estate manager of this initial estate, who's in his 60s and been working with tea his entire life, and most all tea is managed the same in this region. And so, yeah, there was definitely some skepticism, and he's fully on board now, and we're expanding this project, and we're really happy about that. So, just a couple notes about how this went in a remote sense. Um, we do weekly calls, sometimes multiple calls per week. Uh, we managed all the composting through WhatsApp and Zoom and shared Google Sheets. And we just tracked everything so closely, meticulously. I can't believe I could not have anticipated it going any better than this. It's early morning for them and late nights for me on these calls. And with these teas in the beginning, no tea was applied without assessing it first. So basically, it would be early morning for them. I'd come in, the tea would be done. They would share their microscope screen with me, and we would go through and do assessments of these teas before they went out. And that's all gone incredibly well. Again, better than I could have expected. Here are just a few more photos. On the far right, that's a photo from this week constructing new windrows for this expansion and the initial estate as well. There's the initial brewer that we designed together. So there was a lot of back and forth on how to do this, how to what kinds of materials to use, what's the regenerative blower, what's the protocol, what's the clean out process on this, how do we make sure these teas are viable and aerobic, and yeah, so I'm just really looking forward to the next phase of this project. How are we doing on time? I don't have a timer on my end.
We're doing great. I'd say another 10 minutes or so, and then we can hop over to Q&A.
Okay. So I just wanted to impart some sort of observations and strategies of my own over the years. My preferred system for these large managed transition is for there to be an on-farm composting program. It just provides a steady source of crop adapted and associative microbes. And I think it's one of the greatest predictors of farm success, at least from my perspective and on these projects that I work on. I would say, and I think Elaine maybe would say this too, but my overarching observation is if you can make compost in which fungi can thrive, the rest will follow. I think that's always the tough component to get right. We see the nematodes start to show up. We see, we see the protists start to show up. Also very important. Those are just critical microorganisms to have in the soil at the outset of a regeneration process where you're trying to rebuild a soil food web in the soil. You've got to get those grazers in there working on what is usually excessive bacteria, depending on which soil or which region you're in. And then the fungal component is, again, often the most challenging. I think it's the first to vacate due to disturbance and the last to reestablish in soil. So what this looks like is minimizing disturbance and obviously avoiding chemicals, which is not the easy way, as it turns out. Maintaining living roots and plant diversity for optimum microbial function and structure within that soil. An observation that I've made over the last probably 6, 7 years is that you should really be striving for a compost with at least 2 to 3 times the minimum microbial targets for fungi, protists, and nematodes, at least those groups, um, as compared to those minimum numbers that are taught through the school. Yes, start by reaching those minimum figures, those minimum biomass and population numbers in your compost, but really, in my experience, and especially once you get into working on projects like this, you need those values to be 2 to 3 times that amount in order to make a biocomplete extract. And I think a lot of people who've studied with the school, a lot of my peers, other practitioners have seen this as well. If you don't have very robust compost, it's going to be very difficult for you to make a biocomplete extract. It can get to the point where in a gallon of water, you're needing to put 5 or 6 pounds of compost, which is, more of a slurry than anything else. It's basically more compost than water, and that doesn't really work out. There's not a lot of fluid to pull from that. Of course, you can dilute and all that. That's another story. I digress. But um, often the majority of microbes remain in the compost after extraction. And so I would say that microbes are more resilient than I thought. And maybe we're being a little too gentle with some of our hand extractions. And I would urge you, yes, of course, check your compost extract. If you're able to use a microscope, do your counts on that. But go and look at the compost you extracted too. And if you have an initial count on that compost, initial organism count on that compost, what does that count look like after you've done your extraction? I'm not saying there won't be organisms left in that compost when you're done with your extraction. There will be, most definitely, but um, are we getting enough into our extract? And so something else that I've realized in recent years is that mechanical extractors often make stronger compost extracts. And I cannot speak for all of them because I only have experience with a few, but we've definitely been able to push, with some of these extractors, let's say, um, 500, in one case, 500 pounds of compost into 250 gallons of extract of water. And it's the strongest extract I've ever been able to make. Yes, we needed to dilute it in order to apply it, but yeah, I was just kind of blown away. We were obviously working with really great compost, but I think we were in the 300 and 400 micrograms per milliliter of fungi in this extract, and I had not seen that before. So just kind of want to impart my 2 cents on compost and making extract. So crop resilience to herbivory or insect pressure and disease is tied to soil microbiome. There's a lot of research there's a little research that has come out in recent years that proves insects are receiving their microbiome from the soil. And so in some of this research, they look at the operational taxonomic units of microbiology between the soil, between the crop, and between the pest. In this case, it was a caterpillar. And what they see was that although, the genes, the genomic makeup of these caterpillars were the same, whether they were feeding on a plant in an intact native soil versus leaves in a sterile environment, the plants on that intact native soil had 3 times the operational taxonomic units of than as compared to the caterpillar feeding on a leaf in a sterile environment. So again, these pests, all insects, whether they're pests of your plant, which are telling you something, or they're soil mites, detritivores, part of the soil food web, all part of the soil food web, really, they are receiving their microbiomes from the soil as well. And if we look at some further research, Um, they've shown that plants in an intact native soil microbiome saw 3 to 5 times less aphid and flea beetle damage than those in sterile soils. And what they were actually doing here was they were taking wild native plants, they were going out and they were extracting the microbiome around that plant in nature, making a water extract, and then watering it in to that soil where they were growing that plant and doing these experiments. And what they saw again was these native intact microbiomes, soil microbiomes, were leading to 3 to 5 times less aphid and flea beetle damage on brassica in this case. And it was not correlated to defense chemicals inside of the plant, glucosinolates inside of the plant. Having this intact native microbiome was deterring herbivory of these pests, but was not actually increasing the defense chemicals inside of the crop. So it's not about, yes, systemically acquired resistance and all these defense pathways, they're important for sure. We want to bolster them. We want plants to have control over that when they need it, or in our cropping situations especially. But it's that's not what's actually going on. So what it looks like to me is that these insects again are getting their microbiomes from the soil, and the more complex these soils are, the more resilient these plants are obviously going to be, and the less of an incidence of herbivory or insect pressure or crop damage or loss you're actually seeing. So build that microbiome, build that soil food web, as Elaine taught us. And it might take some time to get there, and it might need to be a managed transition to get there. But I've seen it time and time again through my projects. Even if we have some pressure, we always see a reduction when we start to do this right, when we start to get these organisms back into the soil. Typically, I can see the protozoa and nematodes show up the first spring or the following spring after we start our inoculations. Fungi, again, as I expressed, comes a little bit slower, but just seeing those organisms pop up and starting to rebuild that soil food web is just it has such an immense impact. And with regard to nutrition management and trying to maintain yields, actually, we'll get to that in the next slide. So, track your microbial biomass in your soil, or have somebody do it for you. But with that said, expect large seasonal swings in that activity, right? These pools of microorganisms, this activity, it's always in flux. It's changing all the time. There's a big climactic and seasonality component to this. So test your soils at the same time each year. And my suggestion is to do it when you think activity is highest. So if we're talking about the West Coast, I would say when the soil warms up in May, June, that's when I typically like to do my assessments to see what's going on. And then finally, fungi are key players in both organic matter decomposition and humification, which is something, as a compost producer, trying to learn more, doing a lot of DNA testing on our compost and identifying the actual fungal species that are present there, whether or not they're showing up in the soils where we're applying extracts or broadcasting this compost. I'm very interested in this kind of thing, and so I'm typically studying these species that are coming up, these coprinoids, Coprinopsis, a lot of the fruiting bodies that some of you who make compost see. I mean, those, spoiler alert, those are by absolute abundance some of the main species you're going to see in your compost, and they provide a lot of ecosystem benefit. But yeah, so we've been sort of looking at like the top 10 species. I didn't list them out here because I don't think we have enough time to get into all that. But essentially, we think about saprotrophs as doing a lot of this litter decomposition, especially with more complex materials like lignin, lignocellulose, those kinds of things, which we tend to not have enough of in our agricultural settings. So try to maximize that a little bit to have some fungal food out there. But typically mycorrhizal fungi, and this might be changing. There's some research out there that looks like maybe they do have the ability to decompose organic matter and litter in soils. Typically, they're relying on their saprotrophic partners to kind of break things down into more basic components that they can then mineralize themselves and feed back to the plant. But what we see essentially is that, through this humification process, part of what's going on is that fungi are creating these macro aggregates in soil along with plant roots, which entangles minerals and organic matter and protects it. So that's sort of this humus, this mineral associated organic matter, or mineral occluded organic matter, depending on which terminology you prefer. But within these sort of this enmeshment of roots and organic matter and minerals and fungal hyphae, there's a lot of biomineralization going on there too. So not only are they breaking things down, but through the process of humification, they're making things more complex as well. They're excreting polymeric compounds and they're precipitating and nucleating new minerals within those areas where this humification is occurring. And that's a big part of what's going on there. So I don't just think of our saprotrophic fungi or our mycorrhizal fungi, for that matter, as just decomposers. They're actually the ones who are orchestrating humification and this stabilization of carbon in soils. And so, yeah, I'm fairly certain that that information could turn some of these carbon credit buffs into mycologists overnight, but we'll see how that all goes. There's more to that equation with humification. A lot of it is still being worked out. Quinones and their ability to sort of polymerize with sugars and amino acids, etc. But I mean, they are these just, fungi is just this massive component that is missing from a lot of our agricultural soils, and they really are keys to this soil health and stabilization of this carbon, fixing water cycles, holding water, crop resilience, climate issues. I think fungi really is the key to that. All right, do we have one more? How we doing, Evan?
Doing good. We're definitely in overtime, but I think you got one more dense slide, so I definitely don't want everyone to miss this information, and then we'll jump over. We still got about 45 minutes, so plenty of time for Q&A.
Okay. So, yeah, just the managed transition and working with nature. Living soil is a decentralized system of overlapping and entangled feedback loops. So, the context for these managed transition is how how do we improve soil health, microbial diversity, and humus formation while maintaining yields? And so, with heavy nitrogen and phosphorus fertilization, we see decreased rhizodeposition of carbon from the plant, decreased mycorrhizal colonization because these plants have what they need, so they don't, feed their mycorrhizal partners. Decreases in microbial diversity, accelerations of organic matter decomposition. You put high levels of nitrogen in there, it's going to spike activity and respiration. They're going to need a food source to sort of balance out all that nitrogen they've just received. So they're going to go after the organic matter, and that's going to be an accelerated decomposition. Um, decreases humus formation as well, and favors pathogens over mutualists. I was just reading a study the other day where they showed that heavy nitrogen and phosphorus fertilization favors these pathogens over mutualists in grasslands and prairies across 4 continents. And that correlation was very strong through that study. So again, things Elaine has been telling us from the beginning, and more science coming out around this stuff. A lot of this we knew probably for a long time, but the evidence of it is just getting stronger and stronger. So, in a nutshell, just some notes to kind of leave you with. My approach to the managed transition is judicious rates in soil and reducing them over time. If we can go right to minerals and organics right off the bat, that's what I will do every single time. But, I will assuage concerns from large scale clients to make it a slower transition when it's necessary. Again, favor compost manure, amino acids, and minerals when we're talking about nitrogen. Fertigation for efficiency and priming versus front loading. Even when we are using manures, putting 6, 8 tons per acre down in the spring is going to have a very similar effect to what we see above here with this heavy nitrogen fertilization and all of those responses to that. So I prefer to use a smaller amount in the spring, let's say a ton or two, and then use fertigation or coming back and side dressing throughout the season. Let's spread it out a bit. Foliar applications help keep a lot of that out of the soil while you are working on these transitions and maintaining your yield and quality. I also think that nutrient density and foliar nutrition is going to help a lot, yes, with quality and yield, but also just making a plant more resilient. So we need to get good data if we're going to utilize those types of principles. So I work a lot with a SAP lab here in Oregon, Apical Ag, and we just take a very precise approach to it. You know, what's there, what's missing, what's critical for this stage, and we put things together. We chelate them with organics, amino acids, humic acids, soluble carbons, and we see pretty profound responses when we have the data and we formulate these applications carefully and consciously. And then, of course, leverage viable inoculants like mycorrhizal fungi, diazotrophs or nitrogen fixers, sometimes nitrifiers, depending on where you're at in the transition process, and P solubilizers as well. So mycorrhizal fungi is going to do that, but plant growth promoting bacterial strains as well. And that's it. I'm not sure how long we went there, maybe a little over time, but I appreciate everybody's attention and yeah, open to hearing some questions and having some dialogue from here. Thank you.
Thank you, Nick. You just made us, you kept us in wow from the first slide to the last one. And I'm dreaming to when I can go and test that tea as a tea person. I'm just like, my gosh, I need to have that one. One day I'll get there, hopefully. And I think that it's time to go for Evan.
Thank you, Carla, and thank you, Nick, for such a riveting presentation. I have so many more questions, and I just feel like I've gotten such a download that'll take some time to digest all the really well presented and clear information, and just so lovely to hear your story from, the early days and just finding out about Elaine and some of your early education all the way up to present day and the way you've managed to scale that and bringing back some practical insights to the community so everyone else can accelerate their journey through your learnings. So yeah, just want to take a minute to talk about the Soil Food Web Foundation. For those who haven't heard yet, Elaine has recently retired, and this uh, presentation, this webinar series is really all about celebrating her and her legacy and the way that lives on through all of the folks that she taught and touched throughout her over 40-year career. And so we've turned the Soil Food Web School into a foundation and are now carrying on and stewarding that legacy. We've got a special offer right now as a part of this celebration, which is the foundation courses for the lowest price they've ever been offered, along with a cohort experience that we've designed, which is designed to take you through those courses that Elaine produced and have really been the foundation for a lot of this learning, alongside some of the expert mentors from the school, who are usually just teaching our advanced courses as well as a cohort of other peers. And we're also open for donations now as a 501c3. The generosity of this community has really been an important part of us even getting to this point, and is going to be essential for us to continue to achieve our mission and to empower more folks to be like Nick and take this knowledge and put it out into practice in the world, and also to generate new knowledge and even to host webinar series like these. It's an incredible effort that the team takes on, and I just want to give a special shout out to Sammy and Alex in the back who are making sure all this runs smoothly while we're here presenting. So with that, I think we should jump over to the Q&A. Carla, do you want to start off with the first question?
Yes, I will do. Thank you so much, Evan. Uh, okay, so the first question is from Katie. Nick, could you share specific examples for what didn't work in your compost and what was missing? More specific, what issues did you observe in compost on other farms? And finally, what are the key elements for a good compost? How do you properly make and apply compost teas?
Thank you, Katie, for your questions. Yeah, so a lot to say. I'll try to keep it brief. As you can see, I can be a little long-winded. So, um, I think the moisture management is big. I also think that whether or not, I think with smaller piles, it can be difficult to manage moisture effectively, especially depending on where you live, whether it's dry. I think another component is whether or not the carbons, how, let's say, soluble the carbon source is for that fungal component, right? So if we look at sort of the bulk, the access to a wood chip, so sort of like the surface area to access ratio of a microorganism, a lot of the internal parts of big wood chips are not really accessible. So think about that when you're building out your recipes and maybe you're targeting like 50 or 60% high carbon or woody material in those recipes. How much of that carbon you're putting in is actually going to be accessible to those organisms? And is there another source of more soluble carbon that you could also use in conjunction with something like a wood chip to help? So, straw could be a good example of that, one that I use often. So yeah, I mean, issues is just poor management. I think first of all, like these proportions that we're talking about to make a biocomplete compost, let's just generalize and say 10% high nitrogen, maybe 35% green material, and 50-60% somewhere in there high carbon materials. I think that it's just sort of a mishmash of stuff, which I can understand, and you can still make good composts in doing that. But I also think the moisture is not managed. It's not necessarily turned once it gets hot. So you've got a lot of CO2 building up in that compost, and you're not allowing that to dissipate and reintroducing oxygen. So decomposition is not real efficient, and you just don't end up with the full diversity of organisms.
Yeah, I think that you touch on one of the main key points in my perspective as well is moisture. If the moisture is not optimal, the microorganisms don't have how to do their work. So, like you said, this question we can spend hours and hours. So, we can move for the next one. Are you fine, Nick?
Key elements of good compost. I think we kind of covered that, but
Yeah.
maybe just the reason we're putting these high carbon inputs in there is because we need that fungal food there. And I
Mm.
look at well, Elaine probably taught me this, but looking at the compost pile and the various phases from mesophilic through thermophilic and into maturation as a microbial succession. So we start out with a lot of bacterial activity, actinobacteria, some fungal thermophiles as well, creating all that heat and chewing up all those really simple food resources, celluloses, amino acids, nitrogens, these kinds of things. And that allows a lot of decomposition, a lot of breakdown to occur. And so as those resources break down, those simple resources are utilized, that activity slows down, and then only the specialists who are able to break down those woody materials are able to work on what's left. So if we can keep it aerobic through that thermophilic, and we have that high carbon fungal food on the back end, you keep that moisture managed, you will see a proliferation of fungi because they're going to be the last ones left to kind of work on that. And that's the way we really set it up. So we go from, you know, Really simple on the microbial succession side, really early on in succession through like mid succession. And then that maturation stage, the fungi are really going to work on that carbon component. And it's going to be sort of more like that grassland or prairie type of soil environment, if you want to look at it that way.
Yeah,
And then compost teas, yeah, that's a tough one. In a nutshell, less is more when it comes to feeding a compost tea. Make sure you've got good compost, obviously. Lots of different ways to do this. Make sure it stays aerobic, but don't overfeed it in the beginning. And if you feel like it's sort of stalling out, you're using your microscope, you're seeing it sort of stall out around, I don't know, let's say the 14 or 16-hour mark, don't be afraid to come back with a little bit more food at that point to finish out that tea and just see what kind of activity you end up with from there.
that's
Thanks, Katie.
beautiful. And how do you see, to expand a little bit more, how do you see the variation in food types in your compost? Do you vary much, or you have your preset that is your food mix to go?
I follow the proportions fairly closely, but it's different feedstocks in every situation. As we sort of looked at with the India tea, um, I had not worked with any of those feedstocks before, other than cow manure or cow dung, as they term it. Um,
Mhm.
so it's a little bit of everything. I work with a big operation now where we have a composting program, and they bought, new Turner water trailer, all the fancy stuff, and they process on farm and they generate a lot of dry stem material from like perennial medicinal herb crops. And so we're working with, and those are just amazing feedstocks, by the way. If I could put that in all my compost, I probably would. But um, I think it just comes down to those proportions and looking closely at the C to N ratios of each of those feedstocks and seeing where they fit and building your recipe from that standpoint. It's slightly different feedstocks from farm to farm.
Beautiful. Thank you. Evan, do you want to take the next question?
Sure, happy to. The next question is, I believe, from Walt. And Walt's asking, how did you measure fungal and bacterial biomass for using a microbiometer or microscopy sampling?
Yeah, good question. I microscopy in short. Yeah. But we've also looked at PLFA on a lot of farms. We do some DNA analysis as well, where we can at least get, we're using ITS, so it's tough to get absolute abundance, but we get relative abundance, and we can get a fungi to bacteria ratio as well. But for me, the system that works the best, tells me the most, is microscopy right now.
I don't
Thanks,
need to
Walt.
say which microscopy, right? I think at this point
Uh,
everyone knows.
yeah, I think so. I think so.
Awesome. Thank you so much, Nick. The next question is from Alinani. What affordable, locally buildable equipment can be used in rural regions, for example, Zambia, to brew and apply compost tea on a 10-20 hectares where commercial machines are too expensive?
Yeah, it's a good question. For each of these projects, the same question, especially these remote projects, these same questions are asked. And we just have to figure out what kinds of local materials are even um, available to us before we can really start to break it down into, okay, why these tanks versus these tanks, but, simple cone bottom tanks. I think. I think the regenerative blowers are a very expensive component here, especially as you have a larger and larger brewer. So that one, I don't really have enough experience to say what other methodologies could be used outside of using a regenerative blower. I've always had some kind of a pump or aerator to make compost. So that one's tough. My typical response would be to make compost extract instead, to make really good compost and to make compost extract from that and use it perhaps in a similar way you would use teas. Yes, mostly I'm putting compost extract into the soil, but there are occasions where we will do a foliar application with compost extract. And same goes for tea. Mostly it goes on the foliage, but we will do soil applications of tea sometimes as well. So if you can make really good compost, then you can make a good extract. Just don't be too gentle, as I said before. Um, if you have a very, very small amount, 10 to 20 hectares of compost, then I can understand wanting to expand the microorganisms from that compost and a compost tea. And unfortunately, I don't have a great answer for you in terms of how this could be done without a regenerative blower. But yeah, cone bottom tank, regenerative blower, nothing inside of the tank. That's what I prefer. I've worked with some other models, some airlift style as well, but I like to keep it very simple when it comes to compost tea brewers. Um, I don't know. What do you think, Carla? Is there a way to get it done without the blower that you're aware of?
Yeah, I think that it's like you said, is try to figure out what you have available to you. And I think the secret lies on the proper aeration in the system. As long as you can adapt something nearby that you can keep that aeration level to boost up the good biology, then I mean, it's really put the ingenuity to the application. Check the slides that Nick presented to you, the other brewers and equipment we have in many other webinars, and put your mind to think through what you have available. Then it's go to the field and use Elaine's words, test, check under the microscope to see if the biology is properly there. But I agree with you, Nick. It's just like without knowing what they have available, sometimes it's just like we need to talk a bit more
Yeah.
and help them to figure out what can be adapted based on their reality. That's how
Yeah.
I would go for, and it seems that's how you do as well.
Yeah, I would say one more thing about it. I think I kind of missed this, but she's talking about commercial ex machines. So these are like complete brewers that you buy, and they do
Mhm,
tend to be very, very expensive. So
mhm,
we take more of a DIY approach. I buy all the components and we put it together, and we do it for a fraction of the price of a brewer that's already built up and sort of, has a product name and marketing and all that kind of stuff. So we can build something very similar if not better, for
mhm.
sometimes 30-40% of that cost.
Yeah. Alinani, if you want, please email the school. We have some do-it-yourself material that we can share with you, and we're going to give you a nice idea of where to start. Like Nick said, it's just like try to understand what you have available and test it. For a fraction of cost, I think it becomes more feasible. But what I encourage you is to really do a dive in on the material, check online videos to fully understand, I mean, not fully, but to better understand the process. What are the main key points? And then go to start and build your own equipment. And why not even look for local companies that might be open to discuss new alternatives? That's one thing that's feasible, especially with the regen egg growing so much in so many countries. Uh, I would bet that there are companies out there already looking for how can I enter this market. How do you feel about that, Nick and Evan?
Yeah, that sounds great. And yeah, in regards to Zambia, I know I've met some students recently who are doing some incredible work out in Zambia, I think in the sugarcane industry. So maybe we can find some local folks to connect you with. And Nick, I'd love to ask as well. I really love the way you presented a case study from your own backyard and another one from halfway across the world. Could you share anything more with us about some of the key differences that you've experienced in working locally versus working remotely and what kind of tools are important for facilitating that kind of consultation?
Yeah, I would say obviously using Zoom, using something like WhatsApp for international communication, using some kind of a file sharing service, whether it's a Google Drive or something else, and having a microscope on the other end of that remote consultation. That's really big. So though, if you're looking to work with somebody uh, remotely and you're in another country, whatever, a microscope is something that you'll want to think about. And don't be, I don't know, don't be too discouraged by or intimidated by the microscope because we can show you how to get it turned on and to show us what's going on, and that can go a very long way, especially with regard to this composting and compost teas. But yeah, compost tea, I wasn't sure how it was going to work with the India project because I didn't want them just going out and spraying tea that we were assuming was aerobic and had the organisms present. So yeah, I wanted to. I stayed up a little later at night so that I could check those teas before again, we would share live via Zoom what was going on. They would plate a sample. I would look at it. We would count it. They were learning to do these qualitative assessments and are now more proficient at this themselves. So even if you're not quantifying what's going on, it's not that difficult to learn to do a qualitative assessment of these teas. So yeah, I would say that's really the simple equipment to kind of facilitate that, beyond finding a good lab on the other end as well, where you can generate some data to see what else is going on with the soil or crop.
Beautiful. Thank you, Nick. Thank you, Evan. The next question is from Kenneth. Great presentation, Nick. Thank you. I second that. It's an impressive presentation and so many information there. Like Evan said, we need to take time to unpack all of it. How does incorporating mineral chemistry affect microbial functionality? What are your preferred mineral amendments for both cations and anions? And for a commercial farming operation, which compost turner manufacturer would you recommend?
Good questions. Yeah, these are like hot seat questions right here because this kind of thing was not really favored by Elaine, and we had a lot of discussion about this over the years. And again, what we are striving for is a soil that eventually holds and cycles the vast majority of the nutrition biologically. And I think in agriculture, we just haven't seen that. I'm not saying there aren't instances where this is happening, whether small scale or a few large scale, but we're just not there in our agricultural soils right now. So we talk about this, but there aren't great examples of it, at least not across the board. So I think we are still looking forward to seeing the full potentiality of, biological soil benefits in agriculture. So I will just preface it with that. From there, Elaine has talked about calcium to magnesium ratios as one example. And that's because she was over here in Western Oregon. I was over there in Western Oregon. And there's a lot of clay and there's a lot of magnesium. Soils are tight, they're compacted, and magnesium or calcium can help flocculate that. So at the outset of a project, along with some prescribed cultivation, we will be looking at different calcium inputs as that applies. So, whether it's lime, whether it's a lesser amount of gypsum, whether it's something like wollastonite or calcium silicate. Yeah, so those are it could be an oyster shell, which is more of an organic form of a calcium carbonate. So kind of up to you. If you have issues with sodium in your soil already, I would maybe think twice on the oyster shell. But if your sodium levels are very, very low and maybe even that soil or the crop could benefit from having a little bit of sodium there, then oyster shell flour is fine. I would use a lesser amount there. And then as far as the rates, I as far as the rates, I would say that a lot of what is recommended with regard to liming is heavy-handed. I would say a ton or two per acre to start, and then if you're still not seeing your base cation saturation or your calcium percentage get up into the 60s, then you can sort of prime it from there with smaller amounts as you go and you start your inoculation. But I do like to get on that right away at the outset of a project so it doesn't get in the way too much later on. With that said, we talked a little bit about fungal biomineralization and the fact that fungi can both utilize these minerals and they can also produce them. They can produce calcium carbonate, they can produce calcium oxalate, they can produce gypsum even, and they can do this in a controlled or an induced way where they either do it internally or it happens on the exterior up against some of those polymer substances they secrete. So there is always going to be interaction between microbes and minerals. If you look at fungal culture, if you've ever grown gourmet mushrooms, and you've looked at sort of these blocks, look at the amount of calcium that is used in some of these blocks to grow mushrooms, and just use that as a proxy for, what can fungi actually handle? Because to me, it seems like calcium is pretty heavy in a lot of those blocks that are used, and I have not seen any detriment from calcium applications we've made, only benefits by essentially, that prescribed cultivation, getting the calcium in there to flocculate those clay platelets and build a little bit of structure so there's a little bit of air in that soil, sort of like building the house before those microbes get in there to build out the rest of that structure. That's the way I think about that. Preferred mineral amendments, I went through some of the calciums. On the phosphorus side of things, I do use some soft rock phosphate, and it is a mined mineral of low solubility. So we're usually looking at maybe like 3 to 5% soluble phosphorus in that. And there are studies out there, we talked about heavy phosphorus fertilization and what that can do for mycorrhizal colonization, for example. There are definitely concerns there. But there are publications out there that show mycorrhizal inoculation plus small amounts of soft rock phosphate can actually improve colonization of roots in some cases because they can work on that soft rock as a source to mine and feed to that crop. So do use some soft rock phosphate. You're asking specifically about minerals. Definitely use some plant and animal based stuff too, whether it be like a fish bone meal for phosphorus, a lot of calcium in there as well, some crustacean meal, soy feather in some cases. Just depends on the scale, the budget, and what the crop and what's going on with that soil. What else? Sometimes we'll use SOP, sulfate of potash, more mineral amendments. Yeah, bricks, basalt, rock dust. Use green sand quite a bit. Try to be on that sort of like mineral side of things as much as I possibly can before we go over to something like micronutrient sources that are in the sulfate form. And mostly we're using those as foliar applications when we're using something like a manganese sulfate or a zinc sulfate or something like that. And then we're still chelating that with organics. So we're using amino acids, we're using humics or fulvics in those foliar applications to make it a little more approachable for the crop and the microbiology on the surfaces of that crop. Commercial farming. So the composting equipment, and I don't have any financial relationship with this company, but Midwest Biosystems, the Aeromaster stuff, I've worked with a lot. We've used it ourselves in our own compost manufacturing. One of the clients that I work with now has a new Aeromaster turner. I've worked with two other bigger operations that have Aeromaster turners, and I've seen them side by side with other turners. If you're looking for a smaller turner, that's not going to be the one for you. I think probably the Frontier Mighty Mike is the affordable small turner to use, and those are great. Those are great too. And then there's the Bacchus, I think is how you say that. Those are very nice. You don't need a tractor. They're a little bit expensive, but Elaine really favored those. She really liked those machines. So there's another option for you. But I will say, just with regard to the Midwest, When you see sort of side by side, when you've seen like the Frontier Turners and some of the other companies, CompTech, I've seen some of those and worked with some manufacturers with those too, but those are massive. It just seems to me that these Aeromaster Turners kind of cut through the windrow like butter compared to some of the others that seem to churn and grind a little bit on the gears. So that's the one that I prefer at this time. Maybe that changes later on. Thanks, Kenneth.
Fantastic. Thank you for that detailed answer. Next question from Creedmoor is, what is the lightest or easiest setup for elderly growers to use when applying extracts and teas?
Yeah, let's just have it. Yeah. I mean, you could use you could use a carafe or a pitcher, honestly. Small amount, depending on, how much garden space you're working with. I would say a watering can also be a very effective tool. You can mix up your compost in the bucket with water. Let's just say to be safe and to make a nice strong extract, let's say a pound per gallon of compost to water and stir that up vigorously, filter it into your water can, and just water that right into your soil. If you need something a little more involved, you could get a Venturi injector and you could actually basically the Venturi goes between your hose bib and your hose and has is a valve with a side hose that goes into your extract. And as you turn on your hose bib and you're watering, it's actually pulling out at rates you can set. From that bucket of extract and through your garden hose. So, you're watering your garden and putting extract on at the same time. And these little Venturi injectors are easy to use and affordable. I think I have one for my garden that cost me maybe $35 and was very simple to set up. So, from watering can to little Venturi injectors, and then you could use a sprayer as well. You're probably going to get lower rates of extract onto your soil with a sprayer, but you can open up that nozzle as much as possible and try to get as much out there as you can. And then I would just recommend watering that in. So if you're going to spray it onto your surfaces, it's only going to infiltrate so far. So I would come back and water those organisms in further after you make your application. What do you think? Did that answer the question?
Yeah,
Lightest.
absolutely.
Yeah.
Totally. Yeah. Thank you so much. I mean, great questions, one on the top of the other. It's just like, and here comes another one from Francis. I know you talk a little bit about SAP, and how did you use, sorry, did you use refractometers, tissue testing, or SAP testing to chart progress? And can you tell us a little bit more about the process and how you use this information?
How much time do we have? Um,
Good question.
Got about 10 minutes and
okay.
more questions to get through. So,
So I have a refractometer. I used to use it more often than I do now. We use it on some projects and not others. Tissue testing, I've worked with tissue testing a bit too, but my preference is to use SAP analysis, where they're essentially pulling pure cytosol from that leaf, no vascular tissues. So you're seeing very high parts per million levels of nutrients because it is such a pure concentration that they're pulling out of that leaf and testing. Um, I mean, for multiple clients, I receive SAP tests and soil test results all the time for which I'm making recommendations. And I build out programs around fertigation and foliar, uh, basically using, yes, some soil testing, but SAP data. What's going on in the soil? What have we done in the soil? What's ending up in the plant? What's ending up in the crop? if we have high this or low that. I mean, sometimes we see things that are seemingly anomalous. Oh, there's a bunch of calcium in the soil, it's going to all end up in the crop. No, actually, if there's too much, it actually hinders the uptake of itself as well as other cations and anions. And sometimes we'll see maybe there's good microbial activity, a lot of, let's say, metal reducer bacteria in that soil, and they're able to feed plenty of zinc, or there's mycorrhizal interactions going on. They're able to bring plenty of zinc, for example, into the crop that we don't see in the soil. Um, that shows low in the soil. So the sap for me is also just been an eye-opening thing with regard to nutrition management and even the way we sort of like balance minerals as we've talked about in the soil. Oftentimes, if it's an oxidized soil, the calcium's probably not getting in. The phosphorus is not going to get in because it's tied up on the calcium and the iron. So there's just a lot of ways to sort of look at this, but we basically use SAP to see what's really going on with the soil work and mineral balancing we've done, what's actually ending up in this crop, and where are some areas or what are some elements that we can actually bring up in that crop to sort of, let's say, sufficiency levels, but to sort of meet some of the levels of other nutrients there. Um, this is definitely a big topic, and I could go on for a long time about it. But we probably, with some of these operations that are doing intensive SAP testing and have these fertigation and foliar programs, we're testing 3 or 4 times a year sometimes in a single field. Usually not four unless we really want to check our work at the end of the year. So we'll do an initial test. I'll make some recommendations. We'll do some applications. Then we'll do another test, let's say a few weeks or a month later, and we'll just sort of see what happened. Were those effective rates we applied there? Were they not? Was it a waste of time and money? Or okay, can we bump it a little bit? Can we bring it down a little bit? So I like to start slow with lower rates and kind of see what that response is, where that level goes inside of the crop through basically continuous monitoring of sap throughout the season. And that is how I build out these fertigation and foliar programs. We are receiving data regularly and we are making real-time recommendations. Over time, you build a knowing or an intuition around what that program is going to look like. After a couple of years of seeing sort of the same trends, sort of timing on calcium, foliar calcium applications, let's say, and you're going to be able to anticipate that and build out your program around that. We see a lot of the same things year over year. We also see as the soil gets more complex biologically, we can pull away from rates in these programs, and in some cases, ultimately the goal would be to pull away from them altogether. But certainly we see reductions over time, and I use that data to kind of track our progress, and it does correlate neatly with yield and quality as well. So yeah, we get a little technical with some projects on the SAP side, but it helps us with that managed transition that I've talked about. It's just a profound tool for that.
Beautiful.
Next
Okay.
question is
Go
from
ahead.
Mel. She's asking, what do you advise farmers regarding worming their animals? It was mentioned in the chat that horse manure is best avoided, assuming because of worming meds. Any thoughts on that, Nick?
Yeah. Well, I'm not too into the livestock side of things, but I will say that when we're sourcing manure, we do ask these questions. Do you deworm? What is being used? What does that schedule look like? Are there any windows where we can collect manure where we don't think there's going to be residue from dewormer in there? So, yeah, I think it's a concern. I think horse manure is a good example of a concerning manure in that regard. And I would say ask questions and try to avoid that manure that happens, let's say, within the weeks and or month after deworming occurs. And when we have sort of acceded to that and followed that, we haven't seen issues with like not being able to produce, fungal biomass or produce, what have you. And I do use horse manure here in Central Oregon on our
Mhm.
property. It's what is available to me, and we make good compost with it. They do not deworm where I get it though. So, but yeah, horse manure does make good compost, and I've used manure from places that deworm where we avoided those lots of manure right after deworming with success.
Yeah, manure is one of my favorites as well, alongside beer mash. It's just like talk with the provider, and you can take these decisions. So I have one more question, Nick, from Marie. Should we always prefer to make slow transitions when starting to become regenerative, or should we make the change right away, cold turkey style?
Cold turkey on the biocides. That's just what I prefer for my projects. Alternatives, sure. Like I talked about some biocontrols and those kinds of things, but we're talking about with regard to the tea project, region that is just blanketed in tea, and has been conventional for a very, very long time. And there was a lot of anxiety around moving away from these things. And we're a few years in now, and we're doing great with that. It's still something that we have to monitor very closely. We don't want it to get outside of our attention and for something to flare up on us. So that scouting is really important. Let's see. As far as a slow transition, I mean, you just have to observe what's happening, with the efforts you're putting forth. Again, cold turkey, the biocides. If it were up to me, I would move away from synthetic fertilizer right off the bat and move into minerals and manure and other organic and plant-based forms of fertility and cover cropping and getting the residue, all that kind of stuff. Sometimes we're doing that in conjunction with those practices, though, and it's a transition where maybe we go to 50%, and then, oh, we're only making one application of it per year, and we're moving to these other things, manure fertigation, foliar, those kinds of things. So you just have to observe what's going on as you make your transition. What is the right pace for you? But I don't think that it needs to be a matter of losing yield or quality year one. I think we can do that without we can, with some of these tools that I've talked about, but we can make that year one transition look really good. We just have to be holistic in the way we think about it and build out a program that addresses as much of that system as possible. And then if we're prepared, we see good results. As far as making the slow transition, it also depends on the crop you're working with. If we're working with forage, if we're working with feed and those kinds of things that are already not really high input crops, I say you go right off the bat. It's a fast transition. If there are big problems, sort of like what I was describing in the example I presented, maybe you go a little slower with that because you're a little unsure of what's going on and how it's going to go as you begin to regenerate that soil. But just observe as you go and quicken or slow down that pace as necessary. So yeah, some
Beautiful.
changes should be made just right away and others are slower.
Beautiful, beautiful. Like Elaine's words, listen, Mother Nature. She gonna
Yes.
tell you what to do, right?
Yes.
Thank you so much, Nick. Thank you, thank you, thank you so much for this incredible presentation. Definitely, we need to watch this many times to process a lot of information that's been shared in these four webinars so far. So once more, I deeply appreciate each one of you guys that came here to watch this presentation and those that came to share all their experience and knowledge with us. The replays are available in our YouTube channel and also in our community. The links are there on the screen. Now, just to reinforce that we are gathering around for celebrating World Soil Day with a Soil Food Web community house party. If you've been there before, you know how much fun we have, and we delightfully wait for you there next time. We'll be at 9 a.m. Pacific time on Friday, December 5th, which is 5 p.m. London time. Do not forget to adjust your clock and double check your time zone. Nick, I have no words to express my gratitude, but I will pass the word to Evan. He has more to share with us.
Thank you, Carla. It was a pleasure being on this with you both. And thanks for everybody who tuned in. And thank you to Elaine, most of all.
Mm. Yeah, thank you, Nick, and thank you to Elaine for being the catalyst and the reason that so many of us are gathered here having these conversations. That was an absolute masterclass on how to apply the soil food web approach in the real world. And I just really appreciate the level of depth and clarity that you brought to us all today. If you enjoyed this presentation, please connect with us online on social media, check out the website, like and follow us, subscribe to our YouTube channel. If you got value out of the conversation today and think that other folks in your network and in your communities would get value as well, be sure to let them know and share this content. You know, it's free and available for the world. And yeah, we're here to continue to spread the knowledge of the soil food web and continue to build on Elaine's legacy to serve the soil in a meaningful way. Nick, I hope we get to welcome you back to another one of these webinars soon. Carla, so great to be with you here today. Thank you for everyone who attended from far and wide, and we'll see you next time.
Thanks, everyone.
See you guys next time.